All right, our next speaker is Mars Society royalty. She's one of my favorite Martians, Dr. Penny Boston, and she is affiliated with NASA Ames. She's a scientist at NASA Ames Research Center, and she's going to talk to us about astrobiology. And the title of her work is Astrobiology by Any Other Name, The Search for Universal Indicators of Life.
Very happy to be with everybody at this meeting this time. For the last number of years, I've been unavailable for just about every Mars Society meeting because of the time that it was being held. This is a talk that actually I developed a more full-blown version for the American Geophysical Union conference in December of 2019, just before we started to experience microbiology at the viral level in all its many manifestations. So what I've done is revised that in light of the last couple of years and compressed it, taken out some boring graphs and stuff. And I want to talk really sort of beyond the Mars scope. As I have thought about astrobiology, of course, starting with my interest in Mars many decades ago, it's become more and more clear to me that as we're moving out into not only our own solar system, but also beyond, to look at potential life process indicators on exoplanets, that really we're looking at the whole solar system scale. So a solar system is either a life-bearing or a not life-bearing solar system. And we know that we are a life-bearing solar system insofar as we have at least one planet, ours, that can do this. But trying to generalize how we go about looking for signatures of life, how do we expand our scope of thinking so that we don't miss signals because it's very different from our kind of life. So I think most people in this well-acquainted audience will know that astrobiology is a gamish of all the different fundamental sciences, along with a whole load of various different kinds of engineering in order to enable us to actually get somewhere to study various planets in our solar system and ultimately beyond. There is a long history, really, of even contemplating this. And I want to point out a couple of different points at which I think significant advances were made in our thinking. And one was really one that hasn't been as much celebrated as some. This occurred in the 40s and the 50s. A Russian named Gavril Tikhov actually sort of rounded up a bunch of enthusiastic young graduate students, which is a pattern that has been repeated in many other kinds of episodes of study of these kinds of topics. And they spent their time looking at the optical characteristics of terrestrial plants and thinking about the extreme environments that they had access to, and going up there and trying to figure out how these difficult conditions could actually affect what the optical spectra of plants would be, clearly with an eye to figuring out how we would go about looking for life. These themes, of course, have been woven through the decades since. And one notable study, Seeger et al. In 2005, looking at the Vegetation Red Edge, which would be a possible spectral feature that could indicate some kind of biosignature for an extra terrestrial planet. So these themes have cropped up over and over again. The term exobiology, which we still use within NASA for one of our programs, was originated in the late 1950s by Nobel laureate Joshua Lederberg, who should have shared the Nobel Prize with his wife, Esther Lederberg, but did not. And then, of course, other names for this study have cropped up. If you have never read The Star Beast by Robert Heinlein, which is an early 1950s book, it's actually a pretty good book, and it's not as dated as you might think. Even Winston Churchill in 1939 wrote an essay on alien life and are we alone in space. So you can see that these ideas, of course, have been floating around and modeled and remodeled and remodeled until the point at which, in this space age that we find ourselves, we actually have been capable for the last number of decades of actually doing the rigorous scientific work, or hopefully rigorous scientific work, to actually go in the direction of trying to figure out how to look for other life, how to understand it, how to characterize it. Will we find it in our own solar system? Will we only find it in other solar systems at great distance? We start by trying to know about life on Earth, the biology that we have here, and understand the conditions on other planets, both here and elsewhere in the galaxy. And then we try to map the life stuff onto the planet stuff. And that explodes in a profusion of questions and approaches. And so astrobiology is a stunningly broad set of potential studies that cut across all of the fundamental sciences, from physics to biology, perhaps AI, ML, in order to facilitate some of our investigations and so forth. So on Earth, we know that we have really obvious biosignatures, and you can see on the left of the screen, that's one heck of a big dinosaur bone. And so that, for us, in our earthly context, understanding Earth biology as we do is an obvious biosignature. On the right is an image from the Mars Desert Research Hab environs from our 2002 expedition, which was Crew 6, that I participated in. And that red stuff, an iron compound that is on some of the fractures in some of the rocks there, is a very not so obvious biosignature. And it took years to really characterize that, show that there were entombed microorganisms that contained some signature material that showed that they were iron oxidizers and so forth. So on the very large scale, if we go elsewhere and we find big dinosaurs, we're very well prepared. On the small scale, it's more subtle and harder. So I'm not going to go through the details of this slide, but I always take an opportunity to show one of my favorite images from Joel Hagen that he created many years ago. But so we have the life-produced gases like we do on Earth. We have biological molecules, and on our planet, these are sort of gooey organic things. And then we have morphology with all sorts of different examples in there that I've thrown in there. But the point is really the main categories. When we look at our planet and we look at the spectra of the different gases in our atmosphere, we see in the top of this graph, we have all of these split out by methane, carbon dioxide, water, et cetera. When you smush all those together, you get this tremendously complicated spectrum that you see on the very lowest insert there. And so this is our great hope for exoplanet studies, to try to look for life that is chemically and perhaps similar to us in its environment and in its manifestation of activities on its atmosphere. At the small scale, the kind of work that I've done for the last number of decades is the microbial effect on a rocky terrestrial planet, even one with water. And I'm not going to go into any description of any of these things. I just want to dazzle you with the eye candy that on Earth, microbes are manipulating the basic materials of our planet, its basic rocks and minerals, using fluids and using gases and emitting gases. And these processes produce a wide variety of even macroscopically eyeball scale visible patterns, textures, and mineral compositions. Sometimes on Earth, we have come up against things that look like they might have been biology at one time, but when we investigate them, like these examples here, we can often find no other evidence, not isotopic evidence or chemical, geochemical evidence or biochemical evidence or DNA or any of those things. And so we're left only with the morphology. But because we have the opportunity on our planet to study a lot of very different environments, we, the community, are beginning to piece together what is the Rosetta Stone of trying to understand material even when it's only gross morphology and has no other signals in it. One of the revolutionary ideas in the last decade or so was brought to life by Bob Hazen and his colleagues, first published really in 2008 as a full description. The crux of this is to argue that the vast array of known minerals that we have on Earth today, and more are being discovered all the time, actually are a biosignature. And they base this notion in the fact that there are essentially 12 primitive minerals in the early solar system. That's what we see in primitive bodies that we're studying. We're making great strides in studying those. The Lucy mission is about to launch in a couple of days. Very excited about that. But 4,400 known minerals at the time that they published this work, they attributed about 1,500 of those, or less than half, to the presence of the liquid water that we have, and the fact that we have a planet that conducts tectonic activity. And then they attributed the rest due to life. At this point, that gives us another potential key in looking at other planets in our solar system. Will we ever be able to tell about the mineralogy of exoplanets to the fine detail that it would require to interpret that as a biosignature? I think we're not close to that yet. But nevertheless, this is a revolutionary idea in my view that gives us a whole other realm of biosignatures to investigate here on Earth and in our solar system. So crystal biosignatures are very common in rocky environments on our planet. Microorganisms of many, many, many different kinds manipulate elements. They break down rocky materials. They reconstitute them in various forms. Some of the most striking ones I show here. The lower left image is from a natural sample from the environment. And the two images on the right are some of our experiments reproducing some of these crystals in culture with the microorganisms that we've isolated from these environments. We often have elaborate living morphologies that we can see at the scanning electron microscopy scale, as you see here. And these range from the lower left, which is a very biological form, and those forms turn out to be clearly very active, alive microorganisms that produce all sorts of other data that we can detect them by. The upper left shows a remnant that is almost entirely coated in minerals. So these are no longer active microorganisms, and yet they still have organic material in them that's quite analyzable. And then the two images on the right are an enduring mystery that I'll talk about very briefly in a couple minutes. These forms we are finding in many, many different subsurface environments around the world. I've been talking about them for almost 25 years, and we still don't know what they are. So they're my on-Earth paradigm for the difficulty of using morphology to understand what we're seeing on other planets. A few other examples here just to tantalize you. The upper left shows cute little fuzzy guys that are happily alive, but the upper right shows an entirely iron oxide coated remnant of life. The scale at which we want to actually attack these problems is something that I think is coming into its own as we are gaining traction with advances in artificial intelligence and machine learning and morphology and pattern recognition. So in order to interpret an environment like this on Earth with these giant crystals that we've been studying in the Nika system in Mexico for over a decade, we see these big giant crystals. And then at the smaller scale, we see sort of rough surface textures at the finger scale that you can see in the lower left. And then when we actually interrogate this with high-powered electron microscopy, we can see all these fine thread-like structures here. And if you put all those together, you begin to see a pattern of everything from the small scale biology all the way up to what it is doing to the geological environment in which it is finding itself. I think that this kind of investigation is something that our robotic capabilities will be able to interrogate within perhaps the next decade or so. I want to just say 2 words about extremophiles. We kind of fling that idea around all the time. At some level, people almost act like all extremophiles can tolerate everything. And we do have one organism that seems almost like that's true, and those are the tardigrades, the little water bears. But the truth is that even though we've been studying extremophiles since the late 1960s, what we don't know about them is probably most of the story. So we don't really know what the minimum water activity that they can tolerate is. The scale is from 0 to 1, and we think maybe it's 0.6, but there are some organisms that have been suggested to be using water vapor directly. It's very hard to study that. I think that's an open question. One of the things that you can derive from a couple of the pictures that I've just shown you from my work of these fantastic shapes of some of these microorganisms, what are they doing? These guys are living on a really tough diet. They are eating salad and not chocolate cake. So there must be some reason that they're actually making fuzzy things and complicated surfaces and so forth. What is that doing on the microphysics surface level? We really don't have an idea. And then there are multiple tolerances that organisms may have that are synergistic. So the well-known organisms that are highly ionizing radiation tolerant, it appears that they actually benefit from the fact that they were actually trying to become salt tolerant, and that also conferred radiation resistance. So the interaction of these complexities is something that we're just being able to scratch the surface of. But all of that said, everything I've shown you is from an earthly perspective, because that's what we have available to study at the time. How universal can these indicators possibly be? Well, I think that as we advance into the rest of this century, it's not just Mars, our favorite planet in this group, but it's also all these other bodies. And all of these bodies are under consideration. Some have missions that are already approved. Some are in the planning process. Undoubtedly, we will go to these many times over the next 80 or so years, the remainder of this century. And I have, well over a decade ago, sort of divided planets in my own mind into type 1 and type 2. And type 1, we have only one example, and that's us. And this is where the biology on the surface is extremely conspicuous. The atmosphere is conspicuously complicated as a consequence of these living processes. There's many other factors here. But all of the other potential bodies in our solar system, if they house life at all, have to be put in my planet type 2 biosphere category, where they have no visible, obvious means of support on the surface. The gases, if any, in an atmosphere are in what we would consider chemical equilibrium. And this would mean that many of them are in the outer solar system, like the icy moons. This would mean that energy, or at least a large part of the energy, would come from chemical geological sources on the interior of the planet. This makes the whole planet type 2 biosphere category rather cryptic. So where do we go from here? Well, I always talk to my cats about this, and they always have good ideas. Astrobiology as a field is advancing very rapidly in many ways. And some of those ways that we're advancing rapidly is really incredibly refined ability to investigate the chemistry of the environment, the chemistry of living things, the geochemistry and the biochemistry, and how they overlap. There are many other areas, though, that are much more difficult to do. And as we contemplate future space missions, how do we use the wonderful devices that we currently have, but also add to those so that we have a broader spectrum of different data types? I think I'm going to skip over everything except a couple of lines here. Based on my work for 30 plus years in the subsurface, looking at organisms that mostly eat rock and poop out other minerals, there are many unifying themes that don't depend necessarily on the chemistry. And this is a list of some of them. We have very large biodiversity. They grow very slowly. They make patterns. They tend to fossilize themselves in place. And so all of these are potentially signals that may not depend on the chemistry. I want to say one word about biopatterning. These patterns that you see, these complex hieroglyphics patterns, we believe that we have seen them in so many different environments at so many different scales on planet Earth that we think that they are actually a reflection of basic survival strategies, not the resources that the organisms are going after, but the way that they go after them, and it produces these patterns. Maybe something like this could be a biological signal. I want time for questions, so I'm just going to finish with this one last slide. A lot of the upshot that I think matters for Mars is based on our observations of the fact that apparently microorganisms seem to be able to survive in geological environments on Earth, particularly buried ones or subsurface ones, for very long periods of time. This has led us to speculate whether or not the subsurface could act on Earth as a geological genome bank where organisms get buried or trapped, essentially still viable, or some small portion of them is still viable over geological time, and then they can be reintroduced as geological changes on the planet allow reemergence of these organisms. I think this has particular potential application for Mars. If Mars does have a remnant subsurface biosphere still, then it is a different case from Earth because it is no longer experiencing the tremendous amount of geological change that Earth has experienced. And so that would mean that such a subsurface system would have been sealed away largely from the surface for a very long period of time. We don't know that. And so this is one of the drivers that I hope to live for a very long time so that I can see how things turn out. And just ending with one of my favorite comics by Nathan Pyle in his Strange Planet series. Sometimes science is so difficult it makes me sad, but very often science is so wonderful that it makes me happy. So thank you very much.
Thank you so much, Penny. Wonderful presentation, super fascinating, and especially the pictures, the macro ones are just wonderful. So I'll ask you a couple of questions on behalf of the audience.
Sure.
And the first question is from Daniela, and they're saying, origin of life is fascinating. The adaptation ability of bacteria in new extreme environments would show us the new ways how to look for life. Are you planning to look for bacteria which was brought on Mars or Moon via contamination by vehicles and such and learn? I think contamination of planets is a risk, but also is a gain.
You know, the planetary protection issues are very sensitive and very complex. So as we are trying to investigate Mars and other planets for the presence of native life, we don't want to screw ourselves up by making it very hard to tell if what we are looking at when we bring samples home to Earth, or when we eventually have people on Mars looking at samples there, we don't want to confuse the issue with contamination. That said, humans need our microbes. We need them inside and outside of us. And so we have to take our bioflora with us. And so the presence of humans will contaminate at least the area around human habitats. I think that it's very interesting, the question that you raised, in terms of what will happen to those organisms when they get introduced into a circumhuman environment. I think we certainly will be studying that. I think we're already sort of studying that by all of the biological experiments that are going on the ISS, and that presumably will be able to go on, you know, at Gateway, and our opportunity to go back to the moon with Artemis to actually make biological investigations there, so we can see how Earth life adapts to the lunar environment and the microgravity environment. But certainly we will learn a lot more when we study those organisms that we unavoidably take with us. At the same time, of course, we want to be extremely persnickety about making sure that we keep any human-associated microbes away, particularly in the early days, from any potential Martian life samples. And so it's quite a balance. And many committees have written reams and reams and reams about this and continue to do that. And it's of great concern both to NASA and globally to the space exploration community.
Thank you. The next question comes from Will, and they ask, do we expect slower metabolism on the icy planetoids? Would this result in lower doubling rates, mutations, and speciation, or any environmental cases for higher rates of mutation, selection, and diversity?
It's so hard to tell. I mean, some people have made the argument that that you can predict clearly whether or not there is enough energy source on a planet, either by incoming solar radiation or internal geological sources of energy, that you could predict whether or not you would get life. But when we look at examples on Earth, in very, very, very low energy environments, organisms seem to be exceeding our expectations in terms of how long they can hang out there. So the simple-minded concept of minimum amount of energy that an organism needs in the past has been based on what it needs to be active over ordinary time. But I think we need to extend this idea to organisms for which we have paradigms here on Earth. We know that in very rigorous parts of our planet, like the North and South poles, many organisms spend a lot of their life cycle being inactive. So I think that the answer to that question, which we can't give a broad answer to, is going to depend on the exact energy sources and the access of hypothetical organisms on the ocean worlds to those. Certainly the temperatures are quite cold in the regions that we understand, but maybe we would understand that there might be spot sources of energy, geothermal, hydrothermal vents or something like that. So it's really, we're just guessing at this point.
Wonderful. Thank you. And the last question, because we've got 1 minute left. Let's see, all of them. There's a lot of great questions, but I'll ask the last one, which is, what statistical probability would you attach personally that life evolved 100% on Earth? Or I guess that the life is beginning.
Yeah, I don't know. I think that there is some case to be made for at least the plausibility of organisms being lofted from one planet to another within the same solar system. I don't know. I would put that probably in the single-digit probability. Further panspermia, like across the galaxy or something from one star system to another, I think that's pretty hard to figure that out unless you're dealing with an extremely radically different form of life that really is much less fragile than what our life is made out of. So, are there silicon organisms hitching a ride across the galaxy? I don't know. In our solar system, I think, we certainly see that rocks are mixing themselves up across our system. So stay tuned. Hope to live for 500 years. Okay, it's great to see all you guys.
Thanks, Penny. Great to see you again. Thanks for being with us.
Thanks.